Okay, in this video, we are going to discuss about the second part of the skeletal muscle tissue. Okay, so let me share the screen for the second part. So far we studied about the structure or microanatomy of the sarcomere. So we know how a sarcomere look like this one. So just to recap, so this is the A band region, the dark region. These are the light band region. So when these are the Z line, this one and this one. So when sarcomere contract, those filaments, that zone of overlap increases, meaning the thin filament goes in more to the thick filament. So it looks like this. They're going towards this more. So now we are going to study like how this exactly happened. Okay, so now let's go a little bit more detail of the thin filaments and thick filaments. So thin filaments, although I said already that is made with an actin protein, but that is not actually all. There are many more things in it. So thin filament, this is how they look like. So this is the thin filament. That's when you make it big. This is how they look like. So it has, see that tiny tennis ball-like structure? Those are called G-actin molecules. So those are called G-actin molecules. Those G-actin molecules, hold on 1 second, I want to remove that. Okay, so those this is G-actin molecule. Those G-actin molecule together, 300 to 400 of them together make a chain. It kind of looks like a DNA double helix structure. So these tennis ball-like structure, which are known as G-actin, like a globular protein, like a sphere-looking protein. So those together make this F-actin molecule. F, filamentous, meaning long. So this is an F-actin strand. So these are all G-actin molecules. So 300 to 400 of them makes an F-actin molecules. Now, this is the important part. The G-actin, which looks like a tennis ball, one side of it has the active site. One side of its active site. In this figure, they put a yellow color. So if you look at right here, this yellow color area, that's the active side of the G-actin. Active for what? Active for where they bind with the thick filament. So thick filament, which is made with a protein called myosin. I'm going to come back here. So let's go to the thick filament. So thick filament, that's a huge blue color material right here. That looks like exactly this. This is made with a molecule called myosin, protein molecules. So myosin molecules, how they look like? They have an elongate tail and a globular head. So that's their head and that's their tail. It looks like a golf club. So if you imagine a golf club, they have a stick and at the head at the end with a hinge. So it looks like this is the tail and there's a hinge, and that my fist is the head of the globular head of the myosin molecule. So myosin molecule literally looks like this. Now, if you consider this is the tail of the myosin molecule, so all their tail is facing towards the midline, that's M-line protein. So all their tails are this way and all their heads are outside, protruded like this. So if you look from this side, the heads are here. If you look from this side, the heads are here. The tails are towards the midline. So these are the tails. So they create like a structure like a golf club. So remember this. Now, this head, this is the important part again. This head wants to bind with that active site. That head, so if you imagine a tennis ball here with the active side underneath and the head of the myosin molecule right there. So that's a tennis ball on top, head of the myosin molecule. They want to bind. They want to bind, but they can't bind in a normal condition. Why? Because now coming back to the thin filament again, this active side of the G-actin is covered with a protein in this figure that's like a green color protein right there. That protein is called tropomyosin. That's the protein. Tropomyosin covers the active site of the G-actin. Just for your knowledge, each covers approximately 7 active sites. So 1, 2, 3, 4, 5, 6, 7. The 7 was covered by this particular protein. Okay, that protein is called tropomyosin. Now, tropomyosin can be in place with the help of a protein right here. This is like a tiny pin, that brown color protein right there. They put the brown color in this picture. That's called troponin. So basically, if you think this way, let me give you an example. So let's say this is whatever the drawing is. You can't see that drawing. Let's say this is the active site. If this is the active site, this is covered by a protein. This paper is covering that. This is the active site. It's covered by a protein called tropomyosin. So the protein is the tropomyosin. My finger is the troponin. So troponin is this one. This protein is troponin. They're holding tropomyosin on place. And because tropomyosin is covering, you can't see this. Let's say this is the active site. So if active site is covered by tropomyosin, and this is troponin, troponin is holding them, so that the myosin head, which is this myosin head, the head of the golf club, they cannot bind to the G-actin. They cannot bind to the G-actin. Now, why they want to bind and when they can bind, that's what we're going to mention now. So why they want to bind? Because this is important. Once they bind, once that head binds to that active site, they pull. That's known as power stroke. They literally pull them. So once they bind, they pull. So if you imagine this, it's like a tug of war. Once you hold the rope, you pull. So if the golf club head, which is the myosin head, if they bind to the G-actin, they're going to pull. And that pull is causing this. Remember, look at this picture now. This tiny extended part of the blue color line, those are the G-actin head. Once they're binding to the thin filament, once they're binding to the thin filament, they're pulling the thin filament inside, which cause sliding filament. So that's the muscle contraction. So basically this, the G-actin, the globular actin in this picture right here, they have an active site which is covered by tropomyosin, and troponin is keeping tropomyosin in place. The myosin head is this, they're just right underneath it. What they want to do, they have a tremendous affinity towards the active side of the G-actin. If they bind, they will pull. By the way, this binding between the globular head of the myosin and the active side of the G-actin is called cross-bridge formation. This is the word, cross-bridge formation. So once again, the head of the myosin molecule, when they attach to the active side of the G-actin, that's binding. That attraction is called cross-bridge formation. So once the cross bridge is formed, they're going to do a power stroke, meaning they're going to do pull. And that pull causes contraction. So now we know why contraction happened. So why muscle contract? Muscle contract because the head of the myosin molecule, once they bind with the G-actin active site, they pull. And that causes muscle contraction. Now the question is, how they bind, because they are covered. So if they're covered, they can bind. So the question is how they bind. So let's go back to our original discussion. So that was our active site. This is tropomyosin. My finger is troponin. So troponin molecules, this is the one, they are holding tropomyosin in place. Now this is the most important part. Troponin molecule, which is my finger, they love calcium. They love calcium ions. Remember, calcium ions is stored in the sarcoplasmic reticulum. We learned first. Once the calcium ion comes into the sarcoplasm, meaning the cytoplasm of the muscle cell, the troponin goes to bind to the calcium. As soon as they go bind to the calcium, see what happened to tropomyosin? They drop. So tropomyosin moved, active site is now exposed, myosin head can bind. Once they bind, they pull. So let's recap one more time. So normally, the myosin head is not doing anything because the active side of the G-actin is covered. So what happens when calcium comes? As soon as calcium comes into the cytoplasm, this troponin molecule will bind to the calcium. So one more time, who binds to the calcium? Troponin, troponin. So this troponin molecule is going to bind to the calcium. Once they bind, they change their conformation and they move tropomyosin. Once tropomyosin move, the active site of G-actin is exposed. Once it is exposed, the myosin head bind them. Myosin head, this head bind them. Once they bind them, which means cross bridge is already formed, power stroke, they pull, cause muscle contraction. So what is muscle contraction again? Calcium released in the cytoplasm, troponin bind to the calcium, therefore troponin moved, followed by tropomyosin moved, which exposed the active site of the G-actin molecule. Myosin heads bind to that active site, which is called cross-bridge formation, and then they perform power stroke, meaning they pull that towards the midline. So the whole thin filament come towards the midline. So they basically go like this. That's called sliding filament, which eventually cause contraction. So so we know now how contraction happens. So muscle contraction is a result of this pull. We got it. Now what question to answer? The question we have to answer that where this calcium coming from and how they suddenly appeared in the cytoplasm. We know where calcium live. Calcium live in the sarcoplasmic reticulum, in the SR. There are tons of them. But how they suddenly released, that's what we are going to study next. And we are also going to look into how that power stroke happened. So we are going to study two more things now. We are going to study, because we know what muscle contraction is. We know what muscle contraction is. So what are we going to study? We're going to study first where and how the calcium come from. And the second is how that power stroke happened. So let's look at those two. So this we already know. I explained to you. Calcium binds to troponin. We got that because calcium released from SR, sarcoplasmic reticulum. Troponin moves, therefore tropomyosin moves, exposing G-actin active site. Myosin head binds to that active site, called cross bridge. They bend, then I wrote bend, you can say pull towards the H zone, towards the midline, muscle contraction. ATP is required. How ATP is used, we are going to study later. This is the sliding filament theory I mentioned earlier. Remember one more time, the A band remains constant, I band becomes smaller, the zone of overlap increases, and then the H band becomes smaller. Okay, so remember those two things. That's a sliding filament theory. This is where the calcium is coming from. So let's learn that, where the calcium is coming from. So calcium, this is very important. Okay. Now calcium is released based on the signal that is triggered or sent by the central nervous system. So central nervous system, or in short, CNS, send a signal to the skeletal muscle. This is the molecule, this is the cell that sends the signal. That cell is known as neuron. Neuron is the cell or a nerve cell which brings the signal from the central nervous system. Central nervous system composed of your brain and spinal cord. In nervous system chapter, obviously, we're going to know detail about that. So this neuron cell sending a signal to the skeletal muscle. This is the skeletal muscle fiber. So when they send the signal to the skeletal muscle, then only skeletal muscle can work. So that is very important. Skeletal muscle cell is not going to work at all if they do not receive the signal, if they don't receive the signal from the central nervous system. The central nervous system has to send the signal. So the signal, this red color indicate, this red arrow indicate the signal comes. So signal, the fancy name is action potential. So when the action potential arrives from the central nervous system to the skeletal muscle, then only they can work. This junction right here between the neuron and the muscle is known as neuromuscular junction. This junction right here is known as, here's the word, neuromuscular junction, or in short, NMJ. We are going to study exactly what happened at the NMJ right here. So basically, the signal, this picture will describe you in short. The signal arrives from central nervous system to skeletal muscle. At the NMJ, something happens. So I'm going to explain what. The signal continue to the T-tubule. Remember T-tubule, transverse tubule? We studied in the first, like a yellow color rubber band. The T-tubule brings that to the sarcoplasmic reticulum. Sarcoplasmic reticulum release calcium. The calcium bind to the troponin. Therefore, tropomyosin moves. G-actin get exposed. Myosin head binds. Pull, contraction. So we are now going to learn exactly what happened right there. This is a junction we call neuromuscular junction. So this side of this picture indicating the end of a neuron, and this side is the muscle cell. So the blue color side is the end of a neuron. So at the end of a neuron, right here, the end of the neuron become a bulb-like structure, like this. So at the end of the neuron, they become like this at the end. So the bulb. This bulb is called synaptic bulb. Synaptic bulb or synaptic terminal. Synapse, the word means junction. So right here, that is the synaptic bulb or synaptic terminal. Inside the neuron cell, within that synaptic bulb, we have a lot of vesicles. These are all the vesicles. Inside that vesicles, the red dot indicate are neurotransmitters we call acetylcholine, in short ACH. Acetylcholine is the name of a neurotransmitter which is stored within that synaptic bulb. This gap between the synaptic bulb and the muscle cell, which is sarcolemma, this gap is called synaptic cleft. They are not attached 100%, like they are not attached directly. They are not physically attached. There's a microscopic gap. That gap is called synaptic cleft. Cleft means gap. And right here on the sarcolemma, which is the plasma membrane of the muscle cell, we have receptors. This blue color material indicating receptor. Receptors for what? Receptors for ACH, acetylcholine. So this is ACH receptors. These are ACH itself. So this red line indicate the signal arrive at the neuron synaptic bulb. So when the synaptic bulb receive this signal, they release the neurotransmitter via exocytosis. We learned that previously, exocytosis, releasing of molecule. That is exactly what they did right here. So the all molecules came out. So when they come out, they're going to bind with the receptor here at the sarcolemma. Sarcolemma means muscle cell. As you can see, this red line indicating signal. As soon as the acetylcholine bind to the receptor, look, they make this red, meaning the signal is already in the muscle cell. This is basically, you put the password, boom, Wi-Fi, you got it. So now you're connected to the Wi-Fi network. So same thing, the acetylcholine bind to the receptor, the signal is in the muscle cell. Once the signal is in the muscle cell, in the sarcolemma, now that signal is going to transfer from sarcolemma or the plasma membrane via the T-tubule to the sarcoplasmic reticulum. As soon as it goes to the sarcoplasmic reticulum in turn will release calcium. Once the calcium is released, troponin bind with the calcium, tropomyosin move, and we know the rest. So one more time, signal comes from the CNS, central nervous system, to here. This type of neuron, which brings the signal from the central nervous system, we call them motor neuron, M-O-T-O-R, motor neuron. So the neuron that brings the signal from the CNS to the cell here in the muscle, we call them motor neuron. That is why this end of the sarcolemma, we call them motor end plate. So this is the motor neuron. This, my left hand is my muscle. So that's called motor end plate. So that's motor neuron, motor end plate. That gap between these two is called synaptic cleft. Now to increase the number of receptors that are sitting on the sarcolemma, see that this is 1, 2, 3, 4, and so on. To increase the number, the sarcolemma motor end plate makes a lot of fold to increase the surface area. These folds are called junctional fold. Those folds are called junctional fold. So fold at the junction. So junctional fold, what is their function? Their function is to increase the surface area so that more receptor can fit into that sarcolemma area. So once the signal arrives here, neurotransmitter comes out, they bind to the receptors, boom, the signal is on the muscle cell, carried out to sarcoplasmic reticulum, calcium released. Got that. What next? At the synaptic terminal right here, you see that yellow molecule they are showing in the yellow color? Those molecules are called acetylcholine esterase. So this acetylcholine E, the esterase, is an enzyme. So acetylcholine esterase is an enzyme which breaks down the binding between the receptor and the acetylcholine. So acetylcholine esterase is an enzyme which breaks down the binding between the receptor and the acetylcholine. So as soon as they bind, the signal goes in a second, like a fraction of a second. See that? They broke it down. So the signal disappears. Once the signal disappears, calcium is withdrawn from the cytoplasm and they go back to the sarcoplasmic reticulum. Because sarcoplasmic reticulum, when they get close, they said, hey guys, come back in. So the calcium comes back in. Contraction ends. The muscle will eventually relax. So contraction begins with the signal goes to the sarcoplasmic reticulum, and contraction end as soon as this enzyme chop up the binding between the neurotransmitters and the receptors. Good. So this is basically called neuromuscular junction, or NMJ, where this whole thing takes place. So now we know that why calcium is suddenly released by sarcoplasmic reticulum, and where calcium comes out and where they go, who they bind, and what happened after that. Remember the whole series. This is the power stroke. This is the power stroke, meaning what happened when the myosin head bind to the exposed active site. So remember, ordinarily the active site is closed. So active site, the yellow part, is closed by the molecule called tropomyosin. And troponin is the protein that keeping them intact. On the other hand, see this head of the myosin molecule, the head of the thick filament? This is very important to understand. They already broke an ATP molecule. So when we learn that ATP, when they break into ADP and PI, ADP and inorganic phosphate, they release energy. Pay attention to this part. They break them, but they don't release it. So as long as you're not releasing it, you are not using the energy. You're going to use the energy as soon as you release it. So they break it, but they don't release it. In other words, I can say this, they are charged, meaning they are really ready to work. I'll give you an example to understand. It is kind of like, those mouse trap they use. So if you pull that string right here, there's a spring attached, you pull that spring right here, you put a cheese there, and mouse come, and then suddenly this thing snap. So this part is ready to use. They have the energy stored in them. They're not using it until the cheese here is touched. Once the cheese is touched, boom, they snap. This is exactly like that. They have the energy ready. They break the ATP molecules, they have the ADP and PI, so they have the energy ready. They're not using it until they release it. And they are not going to release it until they see the active site. So what next? So right here, calcium appears. As soon as calcium appears, they bind with the troponin molecule. As I explained earlier, troponin move. So when troponin move, the tropomyosin also move. So when the tropomyosin move, now the active site is exposed. So as soon as they exposed, look at that, boom. They extended their head, bind, cross bridge formation. So they bound to the active site. As soon as they bind, what happened next is like a simultaneously, they pull while they're releasing this ADP and PI. So it's like the mouse trap. So once the cheese is touched, they snap. So this cheese is right here, they bind, they exposed, boom, they bound, and once they bind, they pull. So now ADP and PI is released. The thin filaments move towards the middle. Got it. Now what next? Another ATP molecule has to come to make that head ready again. It's like you have to come and put that thing back into the, they are not going to go back, that mouse trap we're talking about, they're not going to go back to the original position. You have to physically come and spend some energy to take it to them back to their original position when they're charged. Same thing here. The ATP comes, the ATP break down into ADP and PI, they are charged again, ready for the second move. This is called contraction cycle, or the cycle of the power stroke. So what I'm going to do, I'm also going to upload a video, because I am sure there are a lot of animation videos available. You guys can find it. I'm going to upload one more video in the Blackboard from somewhere from like YouTube. So you can literally see this. I have seen before, there are some animation videos available. So now we know how exactly muscle contracts. So this slide actually review of whole muscle contraction. So look at that. That's NMJ. That's a motor neuron. This is motor and plate. This is called synaptic terminal. The gap is called synaptic cleft. The red dots are the acetylcholine, which is the neurotransmitter for skeletal muscle. Once they get, so when the signal comes, they're released, they bind with the, so when acetylcholine released, they're binding with the receptors, the action potential of the signal goes through the T-tubule, this is T-tubule, to the sarcoplasmic reticulum. Sarcoplasmic reticulum release calcium, calcium bind to the troponin molecule, move tropomyosin, cross bridge formation happens, muscle contracts. On the other hand, few fraction of a seconds later, ACH is removed by acetylcholinesterase, the enzyme right there. Once the removed signal goes back, so the calcium returns or recaptured into the sarcoplasmic reticulum, active site covered because tropomyosin come back, and then no cross bridge, contraction ends. Contraction ends. But then relaxation is basically a passive return to the resting limbs. Remember, muscles are attached to the bone by a piece of tendon. So when the tendon gets stretched, they go back to their original position, so take the muscle back to the original location. That's what the muscle contraction. Now the last part of this chapter is about what is the like regulation. So regulatory mechanism we are going to study a little bit. This is exactly what I said there. It's written here. So when you study this both slides, you will have an overall idea whatever I just said. It is basically the explanation of whatever I just said earlier. Relaxation. So acetylcholinesterase break down the ACH. So what are the things that limit the contraction? Number one, duration of stimulation. Obviously, how long the muscle is being stimulated. Number two, free calcium in the sarcoplasm, how much calcium is available. And of course, ATP. So if you don't have ATP, no matter what you do, you can't contract the muscle, because remember, you pull once, that's it. You're not going to be charged again if you don't have ATP. So ATP is required for muscle contraction to carry on. This is the regulation. So very interesting. So muscle contraction, now we know exactly how muscle contracts. Calcium comes and the whole thing happens. In the sarcomere, where thousands of sarcomeres are there, we cannot control like how many sarcomeres is going to jump in the action. So that is why it's called all or none principle. Either all of them are working or none of them are working. So if calcium is there, this is basically it is like this is, let's say I'm teaching a class. So when I'm teaching a class, if I talk, either like whoever sitting on that classroom, either everyone is going to listen or none of you are going to listen if I don't talk. So let's say I'm not talking, none of you are going to listen. If I talk, everyone is going to hear me out, okay? So it's called all or none principle. So if the calcium comes, all the sarcomere will jump into action. So we can't control the sarcomere, but we have some other control. That is what we're going to talk. Main thing that we control, the frequency of stimulation. The frequency of stimulation. So the how frequently that stimulation comes. And also it depends on the zone of overlap. Okay, so let's study those two. So first thing is the tension. I mean, tension is a result of muscle contraction, it's going to depend on a zone of overlap. Just look at me. If it is too little zone of overlap, they don't have enough binding. So as if like your tiny end of the tug of war, so you cannot really exert force to pull the rope. Same thing here. If they have so little binding, they can do a little bit. If they are too much, let's say too much zone of overlap, they have no room to pull. Like if you're two opponents, a tug of war, like face to faces like that, there is no rope to pull. But if you're something near the middle, then you can pull them better. That's exactly the case here. So when the optimum zone of overlap is approximately like 2.1 micrometer to 2.2 micrometer, this is where, like 2.1 to 2.2 micrometer, memorize that value. This is where we have the maximum tension. So when the zone of overlap is too little, it's not that much tension. When the zone of overlap is too much, too much zone of overlap, not that much of tension. The tension is only between this normal range and when the zone of overlap is kind of in the middle, okay? So this affects the amount of tension. And what else affects the amount of tension? The frequency of stimulation, like how quickly or how slowly, or what is the time lag between a signal that coming from CNS. So it depends on CNS signal. So let's look at first what happened when the signal comes. So this graph in the bottom will explain that. So let's say this part is the tension, means muscle contraction, more tension formed. And this part is the time, okay? X-axis is the time, Y-axis is the tension. So right here, where the green arrow is, the signal comes. So it takes a few milliseconds to get things happen because there's a lot of biochemical activity. Remember, new acetylcholine get released, they bind to the receptors, then the signal comes to the sarcoplasmic reticulum, calcium opens up, calcium comes and then bind to the troponin. So many things happen. That takes this time, and then contraction happens. This is the contraction part. So this is what the contraction, called contraction phase. So that's where contraction happens. And what happens next? Calcium goes back. Now muscle slowly relaxed. This single cycle is known as muscle twitch. So what is muscle twitch? Muscle twitch is a single stimulation, contraction, relaxation sequence in a muscle fiber. So it's in a single stimulation, contraction, relaxation in a muscle fiber. That's called twitch. Now, muscle twitch could be different for different muscle. For example, your eye muscle, see that muscle twitch is very fast because it's a fast muscle. On the other hand, soleus or gastrocnemius muscle, which are like your calf muscles, those area, so those requires a long, like their muscle twitch is long because they take a lot of time to work on it because they're just mainly for the posture and carrying the body weight, not like your eye muscle, which moves your eye. So they need to do it very rapidly. Okay. So that's the single twitch. Now this thing. Now what happened when multiple signal comes? So follow me here. So if the signal comes here, the contraction phase goes up and then signal ends and the relaxation begins right here and then ends. Now, if a signal comes right there, they will go up again. But interestingly, if you notice here, the next peak is higher. The following peak is even higher and so forth. So every time if the signal is coming, every time if the signal is coming at the end of relaxation, the second peak is higher until they reach as an equilibrium. That phenomena is known as trip. Trip means like stairs. So the repeated stimulation after relaxation period is over, the tension keep rising until they reaches in the equilibrium. Now the question is why? Why it's happening? I'll give you an easy example to understand why it is happening. So let's go back here. So just arbitrary number. Let's say as soon as the signal comes here, 100 calcium ions came into the cytoplasm, 100. That's where it happens like this. Now when they're going back, they're not going back in the equal speed. So 100 came. Now when they're up to here, maybe 80 of them went back. 20 still going back. You know, it takes some time. So 20 still going back. In the meantime, another signal comes. So as if like, let's say you have 20 students sitting in the classroom, the class ends. And let's say 18 just went outside. In the meantime, the second class comes in. So you had 20, 18 went outside, the 20 came in again. So now you have 22 students. Same thing here. So if 100 came in, 80 went back, 20 still there, then 100 more comes in. So now we have 120 calcium. More calcium, more troponin, more exposed, higher contraction. So that's the easy way to understand it. So that's exactly what happened here. So that phenomena is called trip, okay? Now, what happened is when you adding this, like this part is interesting. Now think about if instead of signal coming here, the signal comes right here at 25th seconds. So right here, the second signal comes. By that time, not even 80, maybe 40 of them went outside. Like 40 of them went inside, rather. So now we have 60 available. Remember, total 80, now here 40 of them went inside. So let's say the class ends, instead of 18, let's say 10 students went outside, the other batch came in. So now we'll have 30 students, not 22. Same thing here. So now we have more students. So when that's when even the peak goes even higher. Peak goes even higher, peak goes even higher. So when you're adding this wave, these are like wave, because adding this wave, the phenomena is called wave summation. The word is wave summation, the phenomena, okay? But when you look at that tension, like exactly what happened to the tension, this is like when you're looking at the contraction cycle, like the previous slide. But when you look at the tension rises a lot because every time it comes like this. So the tension until it becomes saturated. Like let's say the room only can handle 70 students. So no matter how many comes, after 70, we can't take more and more students. Same thing here. After some time, we have all sarcomere and 100% calcium attendance. Every troponin molecules is engaged. No more maximum tension is reached. Nothing happens. That phenomena is called incomplete tetanus. Tetanus is the tension. So incomplete tetanus, when we send a stimulation before This is the keyword, before the relaxation period is over. So here it is, relaxation period is over right here, but before it, so I'm sending the stimulation here. So when the relaxation period is over, that's called incomplete tetanus, the tension. But when, let's say I send the signal right here, meaning I don't even allow the relaxation period, then it's going to be like this, because there is no relaxation, so there is not coming back. So they just keep increasing like this until, so let's say 20 students you are attending, I don't allow them to go outside. So another 20 comes in, 40, another 20 comes in, 60, another 10 comes in, 70, we reach to the equilibrium. So we are going to reach to the equilibrium or the saturation point faster, but boom, everything goes like a straight line. That's called complete tetanus, meaning relaxation phase is completely eliminated because the signal appears or signal comes before the relaxation phase even begins. Incomplete is when the relaxation phase is in between, in the middle. Here, we don't even allow relaxation phase. That's called complete tetanus. Now, a couple of other terminology, like how many nerves are involved, okay? So this is interesting. So let's give you an example first, then I'm going to come back here. So let's say, just a simple example. Let's say in my class, I have 5 students, okay? So professor to student ratio is 1:5. Obviously, individual student will get much attention, more attention. And then another class I'm teaching, let's say 200 students, okay? So obviously, in those cases, the attention will be slightly less than this. Although, by the way, for official note, for me, it's always same. Whether it is 5 or 500, I give equal attention to all my students. Keep that in mind. Anyway, coming back to our discussion. So in this case, when one neuron controlling just 5 muscle cells, because they make 5 branch at the end of the axon, at the end of the neuron cell. They're controlling 5 muscle cells. And in another case, one neuron controlling like 50 muscle cells. So we consider that what the controlling 5 muscle cell is more precise. That is called motor unit, meaning how many muscle fiber is controlled by one neuron. How many muscle fibers controlled by one neuron is called motor units. So more motor units means more neuron and less number, the ratio will be less. So in eye muscle, for example, we have many more motor units, meaning, let's say, just completely rough idea, let's say 100 neuron is controlling eye muscles, while 5 neuron controlling your calf muscle, because you don't need to be that precise. But here, you need to be precise. So when you need to be precise, we increase the number of motor units. So motor unit is when a single neuron control how many muscle cell or how many muscle fiber. Okay, that's motor unit. Now, in a group of muscle, these motor units are not like organized, they're just random in everywhere. What does that mean? That means if you look at this picture right here, this picture, different color representing, there are 3 motor units. One is this color, orange color, bluish color, and the purple color, 3 motor units. But it's not that orange color is this side, blue is in the middle, purple is on the side. They are randomly organized. So if you consider like this is this muscle right here, we have multiple motor units controlling multiple things. So body wants to engage multiple motor units when they want to increase the tension. So remember, see the same group of muscles you are using. For example, you sometimes shake hand gently or stab somebody or slap somebody or like punch, the exact similar muscle, but you're engaging more motor unit to generate more tension. So that is called recruitment. So increase in muscular tension by increasing the number of motor units called recruitment. So recruitment is a phenomenon when you increase the number of motor unit to increase muscular tension. So motor unit and recruitment, these are the 2 things you're going to study. And this is different contraction. Okay, contraction is 2 different types based on what is happening to the muscle itself. Okay, whether the muscle change length or whether they don't. Depending on that, one is called isotonic, one is called isometric. Okay, one is called isotonic. In the isotonic, the muscle changes their length. In isometric, the measurement is same. Metric means measurement. In isometric, the muscle does not change the length. One more time. On isotonic, muscle change their length as a result of that contraction. Isometric, muscle does not change their length. So isotonic could be two types, because I'm talking about changing length. Now, when it's just changing length, it can happen two things. Either muscle gets shorter or muscle gets longer. So I'll give you an example. When you pick up something heavy, your muscle right here at home, you can practice this bicep muscle will bulge, like getting shorter. Now the same way, if you're holding it, hanging in your hand, like it is like hanging from your hand, you see that your muscle does not bulge. Same way. So if you pick up a book at home and just fold that book like this will bulge. You same book you're holding, like hanging, like carrying the book as if like this way, you are hanging the book like this, like on the floor, this is not going to bulge. So this is going to be like elongated a little bit. That's what we're studying. So depending on the muscle, if it's shortened, we call concentric. If it elongate, we call eccentric. Memorize that. Isometric doesn't change the length. Like you never exceed the resistance. For example, if I push the wall, I can't move it. So my muscle is not going to change its length, but I'm still working. A lot of muscle force is involved, but I'm pushing the wall, which is not going to, I cannot exceed the resistance given by the wall. Okay, so depending on that. So muscle contraction, again, two types, isotonic, isometric. Isotonic does change the length. Isometric does not change the length. Isotonic, if it is elongate, we call eccentric. If it is shortened, we call concentric. Now, where this muscle getting all this energy? ATP. And ATP we already studied in detail. We studied in detail in cellular respiration chapter that how ATP form in the body. So remember this glucose breakdown in presence of oxygen, in presence of oxygen, this one in absence of oxygen, this is in presence of oxygen, this is in presence of oxygen, we form pyruvic acid, goes into mitochondria, acetyl-CoA, and then electron transport chain results in tons of ATP. In absence of oxygen, they do fermentation, like lactic acid generation, which also results in ATP. That's the ATP source. That's the ATP source. But there is one extra feature here. What happened is ATP, because this takes some time to produce ATP, but muscle wants even more fast energy. So what they do, they convert this, or in other words, I'm going to use the word store. They store that ATP in terms of a molecule called creatine phosphate. So right here, creatine phosphate, it releases the energy immediately that when you need it. So instead of like, let's say this is an example like this. Let's say you are earning. So you work in a whole week, you earn some money. In this case, you work throughout the month, you earn more money. We got it. But let's say you need money right away. So sometimes what people do, they save their money. So the money they earn, they save their money in terms of, let's say you bought a piece of gold. Just an example, you bought a piece of gold. So this is a piece of gold. Now, when you need money, you don't have to work whole week to get that money. Now you can sell the gold and you get the money immediately. That's what the muscle does. So you don't, instead of working to make the ATP, they convert the creatine phosphate into creatine, they have the ATP instantaneously. They have the money instantaneously. The enzyme that does this job is called creatine phosphokinase. I didn't mention in my slide, so please note that. Creatine phosphokinase. In short, we call CPK. Creatine phosphokinase is the enzyme that does this job. Okay, creatine phosphokinase, the enzyme CPK, is available inside the muscle cell. If the muscle cell break open, that enzyme gets into the blood. Otherwise, blood does not have that. That's why when doctors wants to find out any kind of muscle degeneration disease where your muscle is breaking down, they measure CPK in your blood. If they find CPK in the blood, that means something happened to your muscle. If they don't find CPK in the blood, that means muscle is not breaking down. Okay, that is where they get all their energy. This is the muscle metabolism, meaning, you know, the cellular respiration chapter, that is where all the ATP is coming up, and muscle cannot work because remember that power stroke is not going to happen without the ATP. We got that. Yes, of course, we know this. Now the final thing, there are different muscle fiber. Different muscle fiber depending on, like, how fast they can work. So we have three different types of muscle fiber: fast fiber, slow fiber, intermediate fiber. Now let me give you an example first, then you will understand this very like this, okay? So, for example, a chicken, I'm sure that you heard about that they are bird. But I'm almost sure that almost none of you have ever seen chicken flying, okay? Because they need a lot of energy to fly. But if you scare a chicken a little bit, which you should not, by the way, but if you do, they will fly from here to there, like a short distance, they can fly. Okay? Like any other bird, they can fly also, like a short distance. But they need to put a lot of energy in a very quick burst of time because you scared them. So they are going to make a short flight because they need a lot of energy to lift their body from here to point A to point B, but then the quick burst of energy. So their breast muscles, they require fast energy supply. So they flap their wings, they fly. Good. On the other hand, you'll see the chicken, they're walking around the whole farm day long, searching for food or apparently for no reason. So the leg muscle can produce a little bit less amount of energy, but they can work for a very long amount of time. They don't easily get tired, but the breast muscle get tired fast because they cannot fly too long. Now, if you notice, if you are non-veg and you eat chicken, you know the breast muscle is more white and the leg muscle is so-called red fiber. Look at the characteristics now. The slow fiber, which has tons of mitochondria because they generate a lot of ATP for a longer period of time, they contain a red pigment, red fiber, red pigment called myoglobin. Do not confuse with the word hemoglobin. This is myoglobin. Myoglobin is a red fiber, a red pigment, which can temporarily store or reserve oxygen. So this kind of muscle are slow fiber. They're producing energy a little bit less as far as the time is concerned, but they can do for a prolonged period of time because they have their reserve oxygen, because they have a pigment, a red pigment called myoglobin. Because of the red pigment, they appear red fiber. On the other hand, white muscle fiber, which is fast fiber, they have a densely packed myofibril, large glycogen reserve. They have a lot of sugar reserve. Boom, they break. They don't need to create ATP for a very long period of time, but they need ATP now. So they don't care about going to, up to electron transport chain. They need to break down the sugar, need ATP now, and they work fast. And obviously, intermediate fiber is between those two. Okay, so most human muscle obviously followed intermediate fiber. Most human muscle, that's why most human muscle will appear like pink in color, most human muscle. But some area, like for example, eye, they don't have slow fiber. They have mainly fast fiber because you need to work fast. So that's how, our body has a different type of slow, fast, and mostly intermediate fiber. That's it. So that is basically your muscle tissue chapter. So in this muscle tissue chapter, you do not learn identification of muscle. I'm not talking about this is bicep muscle, this is deltoid muscle, this is tricep muscle. That's in the lab. In whatever number of muscle you leave, depend on what college you go. That's in the lab. But in this particular chapter, you are you're learning how a skeletal muscle work. So first we need to know about, go back to my previous recording, like you need to know what are the type of muscle. Then we're going to focus only on skeletal muscle. Then you learn the skeletal muscle anatomy, like the muscle cell anatomy first, like what is sarcoplasm, sarcoplasmic reticulum, T-tubule. Then you should know these are multiple nuclei set, and what is myoblast, what is satellite cells, all these things. Once you know their microanatomy, then go to the myofibril in detail. So when you learn myofibril, you know thick filament, thin filament together, they form sarcomere, which is the functional unit of muscle cell, because based on sarcomere function, muscle cell work. Then you learn what is a sarcomere. So their structure, detail, you learn A band, I band, H zone, Z line or Z disc, titin, zone of overlap, all this, okay? Once you finish learning all this, then you go to the sliding filament theory, like how muscle filament goes across each other so that they cause the contraction. So when you're studying that, you study a molecular level of interaction between thick and the thin filament. Then you study F-actin, G-actin, troponin, tropomyosin. By the way, right now I remember nebulin is a protein. Nebulin is a protein that determine the length of F-actin. Nebulin is a protein that determines the length of F-actin. So I think I didn't mention that time when we were talking about the F-actin and G-actin, but it is like this. I'll give you an example. So let's say you're making a necklace, okay, of a pearl bead necklace. So when the pearl bead necklace is formed, your bead are G-actin molecule. Your bead are G-actin molecule. And the string that you're using, that's nebulin. So if you have this much string, no matter how many beads you have, your necklace will be this long. If you have that long string, and then you can have a lot of beads to put a longer string. So the string length is actually the one that determines the length of the necklace. Same thing here. Nebulin is basically a protein that goes within myosin molecules right here. Let's go back to that one particular slide. Right here, see this in this slide right there, that a little bit purplish color, that string inside, that's the nebulin. So the nebulin, they put the F-actins together. So they determine the length of the F-actin molecule, okay? So that was talking about this, and then you obviously go through the neuromuscular junction. So you know exactly what is happening on the junction between the neuron cell and the sarcolemma, or the plasma membrane of the muscle cell. And what are the molecules involved, how it works, look at this. Then you should go to the contraction cycle, like the power stroke, ATP is required for that. Then this is the summary of those two. And then you read the frequency of stimulation and the optimal length, basically the control of the muscle tension production. You learn twitch, then make sure you know trip, incomplete tetanus, complete tetanus, motor units, recruitment, we discussed that, different type of contraction, isotonic, isometric, and finally energy usage. Mostly we studied in the cellular respiration chapter anyway, but this is where the CPK and the creatine phosphate come into the picture a little bit. And finally, we have a different type of muscle fiber, fast fiber, slow fiber, and intermediate fiber. Okay, so I guess that is it for your second, that is for your skeletal muscle tissue.